Good morning, everyone. Nice to see you here. I feel like it's going to take maybe a little while for people to trickle in. So why don't we start with some introductions, and I will go first. My name is Jessica Guo. I'm a scientific programmer at the DIAG group, which is a data science help group for CALS, although this workshop, and other services we offer are open to people outside CALS too. And yeah, today I'm going to be talking about fitting leaf level gas exchange parameters. Why don't we go around and I'll call on folks and you can tell me, name and affiliation, and then what plant or what plant you're working on, or what your reason is for coming to this workshop. And I'm going to pick on Sebastian first.
Okay, hi, so I'm Sebastian. I'm a first year PhD student at the University of Arizona. So I guess plant would probably be cotton. And the reason for coming to this is because we've been using LICOR for different gas exchange parameters in general, and is just trying to maximize getting as much as we can from that data. In terms of like research focuses, I'm yet to specialize exactly.
Well, congrats on starting during a pandemic and almost being done with your first year. Who's your advisor, or what department and advisor do you
Plant science and Duke Pauli is my advisor.
Okay, I think I saw his name signed up for this too. I don't know if he'll show up, but awesome. Okay. Giovanni.
Hi, I'm Giovanni. I'm like a colleague of Seb. I work in the Pauli lab, and yeah, I'm a postdoc. I started last year in January, and yeah, well, as Seb said, we did some gas exchange measurements with LICOR in cotton, sorghum, and we're planning to do something with beans this year.
Okay.
So yeah, we would like to fit these ACI curves, Racer curves, and like get possibly some parameters out of these curves.
Awesome. Okay, so you guys are doing the rapid method, at least.
Yeah, we also fit some like normal ACI curves just to validate if the Racer can actually be used or not. But yeah, it's a little bit more complicated than we were expecting. So
yeah, so I have to admit, I have not done the Racer method. I've heard kind of mixed reviews of the method itself, but I'd love to know more from your group how you find that. Cotton and sorghum and beans, are those all C3 or is there some C4?
Sorghum is C4.
C4. Okay, so you guys are doing both. Awesome. Thank you. Kinsey.
Hi, everyone. My name is Kinsey Bailey. I am a PhD student in the School of Natural Resources and the Environment, and I am working with Dr. Jia Hu. I mostly study ponderosa pine trees in kind of like the northern periphery of the North American monsoon. And we have actually taken some gas exchange measurements on those ponderosa pine trees. And we've also done it up in Montana. So we're trying to characterize the difference between monsoon dominated ponderosa pine and ponderosa pine that have like mainly winter precipitation. So lots of fun stuff going on.
Are you talking to us from Montana then?
No, I'm actually in Tucson right now, but I do go up to Montana for field work. So
I think Jia told me she was in Montana this week or something like that. So
yes, she is actually.
Okay. Austin.
Good morning, Austin Rutherford. I'm a PhD student as well in the School of Natural Resources and the Environment. I work with Dr. Steve Archer, and I work primarily with mesquites and have collected quite a bit of LICOR gas exchange data on mesquite seedlings more so than adults, but that's the kind of data I'm working with.
Awesome. Emmanuel.
Hi, everyone. I'm Emmanuel Gonzalez. I am a colleague of both Sebastian and Giovanni. I'm a second-year PhD student in Dr. Pauli's lab, and my work mainly focuses on the gantry, also known as the field scanalizer up in Maricopa. So I process a whole bunch of phenotypic data ranging from thermal data to chlorophyll fluorescence. So this workshop is particularly of interest to me.
Awesome. Okay. And then I'm going to have my colleagues introduce themselves. David LeBauer is here, as well as Christina Reamer. So David, why don't you go first?
Okay, my name is David LeBauer, and my job is a director of data science for the College of Ag and Life Sciences. And our collective jobs, myself and Jessica and Christina, is to enable data intensive science within the college. And so we have just started this series of workshops trying to meet the need somewhere in between the core classes and the introductory generic material that's taught for computing and data science, that's available already at the U of A and elsewhere by looking at very specific problems in research such as this. We've also taught last month Christina taught a workshop on NEON data, using NEON data. Before that, generally data management for biosystems engineering. So we're coming up with these workshops to address these specific needs, and also if folks need help following up, these are topics that we all have expertise in. So feel free after this to ask further questions. And also, in case it hasn't been mentioned, we have office hours on Tuesdays from 8 to 10 that are open for anybody to come in and discuss and ask questions.
Oh yeah, David, could you throw the link to that, or the link to our webpage that has that, into the chat, please? Oh, Christina's already on it. Thanks, Christina. Christina, can you go ahead and speak next?
Oh, yeah, sure. Yeah, so I'm Christina. I'm in David's group. I work as a scientific programmer. Yeah, got really into sort of computational tools and training during my PhD. So I have a background in ecology. I'm starting to learn a little bit more about like plant ecology and ecosystem ecology. Jessica and I have been working on a project that involves Lycor data collected on Soteria grasses. Though Jessica's the expert on that, and I'm just sort of the noob learning about it. So yeah, I'm mostly here as like technical troubleshooting and such.
Okay.
And David covered the general stuff that we do in our group. So yeah, all the same things.
And Jocelyn, I'm gonna sorry, everyone else already introduced themselves. Could you just introduce yourself and your affiliation, and what plants you want to study, or why you want to learn about gas exchange parameters?
Hello, sorry I was late. I was having issues with Zoom. But hello everyone, my name is Jocelyn. I'm a first year PhD student in the Brian Inquist Lab in the Department of Ecology and Evolutionary Biology. I'm very interested in gas exchange. I have experience collecting data at the Rocky Mountain Biological Laboratory, specifically looking at plants and their water use efficiency in drought environments. And I'm hoping to go back this summer and measure stomatal conductance and intrinsic water use efficiency as well, along an elevation gradient. So very excited to be attending this workshop. And I'll be working with perennial herbs.
Awesome. Thanks. Well, and just to tack on to what David was saying, not only are we here to help enable your individual research, but also engage in some community building. I hope that now you guys roughly know where your colleagues on campus who have Lycors and are interested, and hopefully you guys can help each other, because hearing some early grad students, I'm hearing some postdocs, so different levels of experience, and collectively we know more than any one single person knows. And I also want to clarify just up ahead, I am not pretending to be an expert on this. There are people who've devoted their entire careers to modeling, measuring and modeling photosynthesis. But that's the whole, like, I feel like the need is like, okay, I know something, I do know something, I'm going to share it, even though I'm not an expert in it. It can be a little bit scary, but I think necessary. So I'm going to post the workshop repo on GitHub. Share that with you. Okay. Can everyone, okay, so if you follow the GitHub link that I just posted, this is for today's workshop, April 2021. And if you want to look at them later, or if you want to follow along, my slides are in the README down below. And so I'm going to share my screen so you can see that. Can you see my screen here? Okay. So the slides are here, should be viewable to anybody at the University of Arizona. I'm happy to make a PDF or something available to someone if there's anyone who's interested outside, but that was the settings that I could figure out with my Google Drive. So without further ado, I'm going to go ahead and present because we also want to get to the actual hands-on workshop part of the workshop today. Okay. So photosynthesis, just to start off really basic, there are 3 flavors of photosynthesis, C3, C4, and CAM. And unfortunately, I will not be talking about CAM photosynthesis today. We will be focusing on primarily C3, but also talking about C4 photosynthesis and some of the differences there, at least from a modeling perspective. As we all know, photosynthetic rate varies, and there are key drivers, key environmental drivers of photosynthesis. 2 of the most important would be light, so that's the energy available to drive photosynthetic reactions, and internal CO2 concentration, which dictates the amount of substrate that can undergo the reaction. And so these are commonly measured curves when people use gas exchange equipment such as a LICOR. I think I heard most everyone here refer to a LICOR. I happen to be wearing a LICOR shirt today. They did not pay me to say that or do that, sadly. But I've used both a 6400 and a 6800. The 6800 makes the there's ways to program the light response curves and the CO2 response curves, also known as ACI curves. Okay, but I do want to back up for a second and explain further why we want to estimate parameters. And so there's kind of 2 parallel reasons I think of as the primary reasons researchers want to do this. And the first is to allow greater interpretation. So we have this, photosynthesis is a chemical reaction. And so we have this model scaffold on which we can apply our data and extract these physiological traits and then compare them. I heard people interested in comparing winter versus summer dominated precipitation regions in one species, maybe different ecotypes. There might be different genotypes of crop species like cotton or sorghum. You might be interested in comparing, as Jocelyn was talking about, response to stressors like drought. You might want to compare before, during, and after events such as that. So instead of comparing rates, in which a lot of things are varying at the same time, it's better to compare the physiological parameters or traits. A second reason you might be interested in estimating these gas exchange parameters is for prediction. So there's a lot of these biophysical models at different scales, leaf scale, plant scale, ecosystem scale, but at the fundamental level, they have these equations in them that need, so if you have inputs of light and water and CO2, you need these parameters, and then it can predict quantities of interest like photosynthetic rate, biomass, crop yield, net primary productivity. Okay, and this is just a picture of a 6800 being used in the field in the sagebrush of Utah, in northern Utah. My field setup involves a giant golf umbrella. In case anybody's doing research in the desert, golf umbrella attached to a wagon was my lifesaver out there. Okay, I'm going to start with the ACI curves. So this is a CO2 response curve, and you can extract certain parameters out of this curve. The CO2 response curve, the measured points are the black points, and it's actually the minimum of these 2 theoretical curves, the red curve and the blue curve. So the red curve represents the carboxylation or CO2 limited range. And so when the internal CO2 concentration is low, that lack of substrate is what's limiting, what's preventing maximum photosynthesis from being reached. In contrast, there's also this blue curve, which represents the electron transport or RUBP limited. So sometimes, so that's in the higher ranges of CO2 concentration. And these curves are a kind of enzyme kinetic curves. Michaelis, oh gosh, yeah, I'm just going to stick with enzyme kinetics curves. I don't want to be wrong here. Go ahead, David.
Michaelis-Menten.
Michaelis-Menten. I was like, I'm pretty sure that's it, but what if it has momentary, like, oh my God, what if I'm wrong? And so this isn't exactly slope or anything, but it's a parameter that dictates this curve. So that's VC max is very important, primarily because at least under natural conditions, you would imagine most often internal CO2 concentration is usually less than 400. On the upper end, the defining parameter is Jmax, maximum rate of electron transport when RUBP is limiting. So that's another important parameter that gets estimated. Okay. Now I'm showing a light response curve, irradiance on the x-axis, net CO2 simulation on the y-axis. There are a couple features of interest on this curve. First, there's a light limited region, low light, and then carboxylation region, limited region once a certain light level has been achieved. And for models, things that might be of interest to compare are dark respiration. So once you've dark adapted leaves and measured the net, it's going to be negative, right? So CO2 is actually going to be produced instead of removed from the light core chamber. And this dark respiration is this intercept here. So when there's absolutely no light, how much CO2 is being respired by the leaf. Another very important parameter of interest, it's represented here by the Greek letter phi. Sometimes you see it as the Greek letter alpha, and it's called sometimes quantum yield, sometimes quantum efficiency. This is one thing that makes reading these papers and interpreting some of the symbols a little bit challenging from time to time. Here I'm going to stick with phi, and it's the slope of this initial portion of the light response curve. So the rate of CO2 assimilation, how much does it increase per unit irradiance increase? There's other parameters that might be of interest, light compensation points. So at which point does this curve cross zero? You might, yeah, there's a curvature. This theta here represents this curvature parameter. So there's always like more detailed and specific things that you might be interested in, and the kind of a general. And I'm here today to try to present some of the more general parameters that are common across many models. Okay. So that was C3 photosynthesis, starting off with the basics. Here I'm showing a comparison, a kind of generalized comparison between a C3 and a C4 species. And then the light response curve here on the left, you can see that the shapes are generally the same. This curve's a little bit, you know, it's a little bit higher for C4 than C3. But the shape of the curve is largely similar. That is not the case for the ACI curve. And so notice too that the range here is much lower than the CI. So we're focusing here on the more CO2 limited part of the curve. And whereas you get the single curve in the C3 case, this is kind of more of two pieces, kind of piecemeal fitting together for the C4 species. And again, the C4 species, there's the carbon concentrating inside the bundle sheath cells, so there's higher photosynthesis at lower CO2 concentrations in a C4 plant. So the model that I'm going to present here is the Collatz 1992 model, and it's fairly straightforward, actually. It's the minimum of these three pieces. So the light response curve, this JI piece, the parameter of interest again here is phi, or quantum yield. The second portion is this CO2 limited. Again, the CO2 limited phase in a C4 plant is much lower and smaller than in a C3 plant. But K, which is a new parameter, haven't shown it before, is the initial slope of that CO2 response. And then there's the not CO2 limited portion, and that's described by this kind of flat. So VC max here is the maximum carboxylation rate, but it's pretty steady. So you can kind of increase CO2 beyond that, and it just kind of chugs along. So it's kind of is represented here. And so the total net photosynthesis is described by the minimum of these three pieces of the curves, and then subtracting off, of course, the dark respiration or the intercept of the light response curve. Before I move on, are there any questions so far? Okay. I'm going to move on to stomatal parameters. And so, as you guys know, photosynthesis and stomatal conductance are intimately, integrally connected by the stomatal aperture. And so what affects one affects the other. So just like photosynthesis, stomatal responses are also affected by light, CO2, and temperature. But additionally, they're also affected by that moisture gradient between the inside of the leaves and the outside of the leaves. And here represented as vapor pressure deficit. And so and the other thing I would say that is different between photosynthesis models and stomatal conductance models, what I've presented so far about the photosynthesis model is the classic Farquhar von Kammerer-Berry model, FVCB, you might see it abbreviated as in papers. And that's, like, from what I can tell, pretty generally agreed upon. Everybody, like, people have different versions of that, different levels of intricacy and detail, but that's the structure. It is less so that way for stomatal models. Stomatal models are much more empirical or semi-empirical. It's kind of based on the shape of the data that you collect, rather than necessarily a lot of a priori theory about how the enzyme kinetics works. And so there is not one single stomatal model to rule them all. There's kind of like over time, different iterations have been developed and promoted and integrated into big models. Okay, so these are three of the most common ones that you might see in the literature. This classic Ball-Berry is the oldest, and it's got this linear format, right? This is GS, or stomatal conductance, is a slope times some variable plus an intercept. We all know that form. And in here, you'll see the A net, so photosynthesis is represented. So that's how this model is capturing the response of stomatal conductance to CO2, light, and temperature. It's contained within the estimate of AN. And then the Ball-Berry version has H, or relative humidity. And now conductance actually doesn't scale very linearly with relative humidity, and it does more so with D, or vapor pressure deficit. So that was one improvement. This is the BBL, or Ball-Berry Looning model, here in the middle. And it's still got an intercept, still got a slope, but then it has this denominator part, which is a bit more complicated. And Looning added a third parameter, D naught. This can be problematic. D naught is highly correlated with M. So they covary, so it's very difficult to estimate independently. And that's a problem. And many, like, global models tend to fix D naught at a certain value and say, give me M when we set D naught to 1 or D naught to 1.6. And then finally, there's the Medlin model. Again, it uses D instead of relative humidity. And it's not quite linear anymore. But all these three models have in common is a stomatal slope term represented by the M, and an intercept term, G naught, which represents cuticular conductance. So when there's no photosynthesis happening and the stomata are presumed closed, how much water is leaking from the leaf through the cuticles. The kind of history of stomatal conductance models was reviewed nicely in Minor et al. 2017. Okay, so that's the theory. I'm glad I didn't take too much time on that, but feel free to chime in or ask questions. I or David can hopefully answer some of those questions. But moving on to okay, so that's the theory. How are we going to get these parameters from the data? I got this sheet off of my LICOR, and I want to estimate parameters. There is also, just like this whole ecosystem of kind of models, there's also a few different software packages that can be used to do this, depending on what you want, what you have, C3 versus C4. So for C3 plants, there's the most options. There's the earliest plant ecophys package by Remco Duursma. I think it was published in 2015. You can use this fit ACI to get C3 parameters for the ACI curve, but it doesn't have a function for the light response curves. And it does have this fit BB function for stomatal parameters for all three model flavors that we just described, the Ball-Berry, Ball-Berry Looning, and Medlin. This is a more recent package just called photosynthesis. And it has everything that Plant Ecophys does, except that it also has this light response curve to get these other parameters, Phi and RDark. Finally, there's this Pecan. I don't know if you've heard of Pecan before. David was very strongly involved with it from the onset. It does this is a module of Pecan that does Bayesian estimation of the CO2 response and the light response curve simultaneously. And you get all the parameters kind of in one function rather than two functions. Today in the scripts I'm going to share, we're going to use the photosynthesis package for the CO2 and light response curves just because it's nice to have those together. And then I'm going to show the Plant Ecophys package in the stomatal conductance estimation or stomatal parameters estimation. The fit A, you may or may not get to the fit A because it's a little different from the other packages. So for C4 species, unfortunately, there's fewer things available to estimate those parameters. What there is BioPro for estimating VC max and Phi. It doesn't estimate K and it doesn't estimate RDark, unfortunately. But you can use this optimization C4 photo or opt-C4 photo function in that. And then the stomatal conductance is the same. So you can also use either of the other packages to estimate stomatal conductance for your C4 species. So, and that's from the package BioPro. BioPro is also a prop model. I mean, for C3 and C4, but it's one of the few things I found out there that had a way to estimate C4 parameters. Maybe this is a niche for you guys. Maybe you guys will be the ones to develop the next package for fitting C4. I don't know. Okay. I want to give a shout out. This is a very recently published paper. David's on here actually. A reporting format for leaf level gas exchange data and metadata. As you all know, publishing nowadays, a lot of times you're required to publish your data too. It's less useful for data reuse to just have a CSV or Excel file uploaded. So this is think about it now before you're publishing your data and you're publishing your paper, how you would get the metadata while it's still fresh in your head, basically. Record this and put it in a format so that future you will think past you for providing all this and making it easy in those final few steps to getting from manuscript to publication. And then a couple of additional resources. I chatted earlier with, who did I chat with about this? Giovanni, I believe. We were chatting about the rapid ACI curves. So this is a package that was developed for that. And then there's the PCNE published these kind of standardized Excel methods. I actually haven't looked at those before. Maybe some of you have had experience with these Excel fitting parameters. But just as a full references, here are other things you could look into as well. And then have some references here, the full citations in case you want to go back and read any of those, consult any of those. Oh, someone's waiting to come in. I'm so sorry.
They just were in the waiting room like a second ago, so I just added them.
Thank you. Okay, so that concludes the presentation portion of the workshop today. Any questions so far? Hearing crickets. Okay. Well, not to put you on the spot, but Natalie, I believe you're the most recent person to join us. Do you mind giving us a brief introduction of your affiliation and maybe the plants or systems that you're working on?
Yeah, hi. Sorry, I thought we started at 10:30.
That's okay. I mean,
I guess that is wrong. When did you start?
10 o'clock.
Oh, okay. Dang it. Okay. Sorry. I'm Natalie Melkonoff. I'm a PhD student at the university, and I'm based in Phoenix at Desert Botanical Garden. I work with milkweeds and monarch butterflies.
Oh, that's so cool. Who's your advisor?
I work with Katie Prudick at U of A and then Kevin Holteen here at the garden.
Oh my goodness. Kevin is my close collaborator and was on my PhD committee too.
Perfect. Very
Awesome.
cool. Sorry, I'm late. I totally did not mean to be.
That's okay. That's fine. I'm glad you found us. And then Dr. Pauli, could you introduce yourself as well? I think several of your lab members are here.
Sure. My name is Duke Paul. I'm assistant professor in the School of Plant Sciences. Do a lot of research up in Maricopa, focused on various crops, including C3, C4, so cotton, sorghum, peas, sunflower, a whole bunch of them. And so, yeah, we're collecting a lot of these data, and I guess I just wanted the members from my lab to know what to do with it. Giovanni's been working with Joseph Stanzano and David Hansen at New Mexico about the racer curves for C4. So we were just kind of interested in that and learning more.
Awesome. Well, yeah, I'd love to hear more about that. Is that racer method, is that developed primarily for C3 then? So far?
Yeah, so they only published it on the C3. So Joseph was working on it for C4. We were providing him the data, but unfortunately he took a temporary position in Canada doing policy work. So we were kind of patiently waiting for him to return.
Yes. Okay. Hopefully there's not mass brain drain to Canada in our futures. Okay. I think that's still everyone. And I just want to reiterate for the newcomers that I'm hoping that to develop informally a community of people who work with LICOR, work with gas exchange curves, can help each other. You know, that's sometimes your best resource is each other. And so meet and mingle, or take note of who you might want to hit up for any shared information in the future. Alrighty, Christina, what is the status of our binder?
Okay, so I was trying to set up a really nice environment that everyone could have used that already had all the R packages and such installed, so that we could avoid, for folks who want to follow along, that we could avoid a lot of the installation problems that can happen. I have a working instance of the binder from this morning, but the photosynthesis package will not work and the file paths have not been updated. So most of this is up to you, Jessica, if you want to try to have folks use the binder instance, or if you want to work locally and have people try to install stuff, or just watch you work through this. So sorry about this not working. It was Binder's fault in my defense. Something's wrong on their end.
Sometimes new technology, it's great until it doesn't work. And you have to figure out why it doesn't work. Okay, well, let me just take a survey. You guys can type in the chat or just shout out and unmute yourselves and chat out like R. Are you guys all already pretty familiar with R in RStudio?
You might want to have people use the reactions if you want to have people thumbs up or something. That way it's easy to see.
We're already using R, and so okay, that's a good majority of you, and even more of you. Okay, fantastic. Then I invite you to open up your own. Oh, I guess. What about GitHub? Do we want people to download the data manually or clone it?
I think people could do either. If people are familiar with Git, they can clone, but you can just download the entire repository as a zip file to get the data and the scripts.
Yes.
So if you want to walk people through that process.
Let's do that for now. So go ahead and open up your RStudio instance. And here, I'm going to do it on mine too. I don't have it on my local machine yet, so this will be fresh. Where'd it go? Okay, so this is my RStudio instance. And over here in the perfect. In the terminal window, I'm going to switch over. I'm going to look for where so this is the part that we're hoping to avoid, but everyone's computer is going to be a little different. Your file paths and naming schemes are going to be a little different. So I'm going to CD into my projects folder.
It might be easiest if you just download the zip file from GitHub instead of Git cloning.
Oh, okay.
In case people are not familiar. Yeah, so if you click there, yeah.
Okay.
So people go to the repo.
Yeah, let me drop that in again for the new folks. Oh, that was to David. I don't mean to send that to David. David knows where that is. Here, everyone. This is our group's work. It's public. It's our workshops repo. And so you can go ahead and download as a zip file. Christina, I've never actually done this before, so perhaps you could narrate me through what needs to be done here.
It should just download a file to your downloads folder.
Okay. And then do I open it up or do I just save it somewhere?
Well, it should be saved. You're going to want to go to it to unzip it.
Okay, go down, extract. I do this. Okay, I'm apparently unable to extract this.
No, it is. It's open.
It's
Isn't
extracted?
it? Yeah.
No, usually it extracts and oh, it came. Sorry, double screens. There, here we go. I'm going to extract it to a place where you can find it again. Name it something sensible, perhaps. Okay, great. And now I'm going to navigate it here. And there's the data. It's in this gas exchange folder.
Okay, so you might want to wait a minute and see if people who are following along have gotten to this point.
Yes, can I get a thumbs up for anybody who already has the folder extracted to some location? Okay, got a couple thumbs ups.
And if you need help, feel free to direct message Christina or I. We can help you on the side as we go through this.
Mm-hmm. If there's any like super like blocking thing, can get put into a breakout room, and this whole thing is being recorded. Oh, I forgot to say, this is being recorded. Yeah, I'm sorry. I think we need to yeah. But hopefully you pressed yes and it warned you that it was being recorded when you signed on. Okay, so I'm going to start a fresh script. And I need to set the working directory, right? So I'm going to choose the directory. So this again will be different for yours. Wherever you just unzipped it to, go there. And then set inside the gas exchange params folder, set that as your working directory. Wow. My RStudio is being not great. Why didn't it like that? Okay, hopefully that will take care of that. So just to explain a bit further, there's two folders in here. There's a scripts folder where I have the complete scripts. So if you're following along and you like couldn't type something or I'm going too fast, first feel free to let me know that I am going too fast. If you don't feel comfortable saying it out loud, type it into the chat. And second, know that the full scripts with all of the stuff is available to you at any time on the GitHub repo. So it's okay if there's something you didn't catch. There's this data folder. Is this terribly small? I think it's probably kind of small.
To make the window bigger for all of the RStudio stuff, go up to preferences under RStudio or file or something. Oh gosh, you're on
I've done this before and now I can't remember how to do it. View, zoom in, yeah, Control++.
Okay, that works.
Okay, that might be too big, but anyhow, in this data folder, I have some ACI and AQ curves. We're going to be doing, so ACI curves, I have a C3 plant, Pinus ponderosa, actually, so one of, I think, Kinsey's species, right? And then a Ceteria viridis, so a C4 grass in here as well. And then AQ curves, I have for the C4 grass. Then for some model conductance, I have some sagebrush. And oh, I did not say this earlier during my presentation, but you don't collect gas to model conductance data in the same like ACI AQ curve way that you do for photosynthesis parameters. So we can talk about that when we get there. All righty. So packages we will need, the plier. Again, if there's any of these that it doesn't load, then you have to do install.packages, whatever the package name is. So if there's something that's new to you. We also use ggplot2. And then we're going to use this photosynthesis. So this is a package you might not have that you might need to install. Oh, and I don't have it. So great. This is perfect example of needing it. And then wait for it to install. Okay, so once it's installed, then I'm going to go ahead and Control-Shift-C and comment it out. We only need to install it once, and then library, use library on it. I'm just going to, we could do this manually, but there's a package called UD units. So this part can be optional. That's to convert units. There's a place in here we're going to convert Celsius to Kelvin. And yes, you can just add, what is it, 273.15, I think, to it, to your numbers. But especially if there's more complicated conversions, it's good to use this package to do so. Christina taught me this, actually. I didn't know about it previously. And then for C4, we're going to load up BioCro. So the C is capitalized, it is case sensitive. No package called BioCro. So I'm going to.
I put the installation instructions for BioCro into the chat because it's not, you can't use install.packages to get it.
Oh, I guess I forgot that. Oh, right. This is on GitHub. So packages on GitHub. DevTools or remotes, : install _ GitHub. Okay, let's do that. And there is another unfortunate piece of minutia that I think we need to do, which is this function here. If you can copy, put it in the chat as well. If you can copy and paste that to the next part of your script. Using the tidyverse is great, but it puts things in tibbles. And tibbles are easy for lots of things, but a lot of packages also don't work on tibbles. They only work on data frames. So sometimes you need to untibble things. You like manipulate things. It's great. Tibbles are great. And then you untibble them in order to put them into these functions. Not yet. Okay. I'm going to paste that in my code as well. And it's just short enough that I kind of copy and paste it from code to code. You could like put it as a separate function and use load on it, or use source on it as well to load it as a function. Okay, so I've installed DevTools, install has run. I'm going to comment that out. Go ahead and do library BioCro, and I'm going to run this function so that it'll take tibbles and make them just plain data frames. All of that to get ready to just import data. I'm going to try to keep comments here so you can follow along. So the first thing we're going to do is import C3 ACI. Is everybody with me so far? Could I get a show of thumbs ups for if the package is installed and functions copied over? And if there's also, I think, the slow down option, feel free to throw up the slow down option too. Okay, I'm getting a trickle of more thumbs ups. Excellent. Okay, I think we're there. So one problem, again, this is a 6800. I'll type that in here. Format, and I have looked, there is not a maintained, well done function out there for just reading in the 6800 format and just having things ready to go. It's a little bit piecemeal. So at this point you have two choices, and I will show both choices. One choice is you go into your Excel that you've taken off the machine and you clean it up. You take off that heading and you save just that first tab as a CSV and you read it in as a CSV, and it's straightforward. But you do have to go in and do that for every single one of your sheets, and you might have, I don't know, tens, dozens, hundreds of these sheets, and you might not want to do that, or you might make a mistake doing that. So what I want to show for this one is that I've kept the heading, but we're going to have to skip some lines, but just for practice. So let's see, let's call this foo. This is just to see. I'm going to use the read underscore CSV function. And then remember, our working directory is here. So we're going to go into the data and then into ACI. A and C are capitalized, I is not. And then the name of the file is PIPO underscore 2019-08-3 CSV. So I basically know this is not going to work and it's not going to like it. I'm going to take a look. Yeah, this is the stuff when it comes off the machine. There's constants and headings and things it tells you about, I don't know, what kind of chamber you had on the Lycor setting and all kinds of things. So what do you actually want? I think what you want is one of these, I think line 13 here is what you want. And you want that, and we're going to read it twice. We're going to read it once in for the column headers, and then we're going to read it once in. So line 13 here is the column header, and then the 14 is the units, and the data doesn't start until 15. And then you have to offset that in your brain because it didn't take into account the very first line. Basically, there's a little bit of trial and error, but once you figure it out once for the way your sheets are, you can then automate this reading in process for all of the other sheets that you have that are in this format. So it is worth, I think, the pain of doing this manual way the first time. So first I want the column names. I'm going to call that CNames. And I'm going to do exactly what I did before. So I'm going to copy and paste that, except I'm going to add some arguments. I'm going to do skip. I'm going to skip rows. And so it's on line 13. It showed that it was on line 13, but in real life it's on line 14. So I'm going to skip 13. And I only want that line. So n max, I want it to be 0. I don't actually want it to read in any more data. So let's see what this does. Oh, it doesn't like that. Okay, this is where I'm going to use the piping and just get, so this is because it's empty actually. I used n max equals 0, so it's empty. I just want to take the column names. And I think in the dplyr world, it's just called names. So if I do that, CNames is now a vector. So I use str on CNames, and it's a vector of the actual things I want my column headers to be, all 153 of them, apparently. So this piping, is that new to anybody? No? Okay, so this is dplyr. It's saying do this and then next do this. So lets you string together different commands. And names, I mean, you can do question mark names to figure things out, but it gets the names in that. So I wonder if whole names would have worked. Okay, see, whole names would have worked too. Names or whole names. Okay, now we're going to read it in for real. And now we're going to call it something. So we're going to label it, because we have lots of data sets. So we're going to label it C3 and ACI. That's going to be the name of this. Now we're going to go back and copy this, at least this first read R line here. Again, this time we're going to skip 15 maybe? I can't remember. So let's try 15. And then we're going to tell its column names to be what we just read in, the C names vector, and close that up. Okay, I'm going to run this. I have a feeling maybe that isn't quite right. Okay, that seems right. Yeah. Okay, so we skipped 15 and we came in, and now we have 14 rows and the columns are correct. Yes. It's a lot of messy code for doing, reading in 14 lines of data. But again, if you only had a few of these, manually going in Excel and like deleting the headers might make sense. But if you have more of these, and just it gets to be a real pain. Somebody has to go in and actually do it and not make any mistakes while doing it. So I present this code here as a way for you to use this method, even though it's messy, and you might have to adapt it to the exact shape of your like or Excel sheets. Okay. The next thing that is important that we do is plot it. So this is an ACI curve where I'm not going to make a fancy plot, but I am going to use ggplot and put in the C3 data, the C3 ACI data set. And we're going to put CI on the x-axis and ANET on the y-axis. So it's CI and y equals A. And I'm just going to show it as points. So I'm going to use geom underscore point. And that's it. This is really the most basic graphic. That's what it shows up on my plots window down here. And that's a pretty respectable looking curve. You know, obviously we can debate, and I think different people have different methods on whether you would clean any of this. My instinct would be to leave it in. These are real data points. I don't have any particular reason to suspect that that one's wrong or any wronger than maybe that one. So I would just leave it in. Yeah. Any questions here? Or needing me to slow down or speed up? Maybe you can also ask me to speed up. But this plotting is always worth doing, right? There could have been anything. You could have accidentally logged an extra point. You could have left the valve open and not discovered it till after the fact, which always sucks, but can happen. So always plot your data before you run any kind of routine or fitting algorithm to it. Okay, so now we're going to use the photosynthesis package, and the function I'm going to do is called fit ACI response. And I'm going to look up what it needs. And so, you know, just every package is a little bit different. So we have to see what it needs, and R makes it easy to look up the functions. What are the arguments? So it needs the data, and it needs a list of the var names. And this is important, right? Because the var names, the column headers change between the 6400 and the 6800. And so if you have 6400, you would be putting photo instead of A. But if you have the 6800, you're using A. So you're gonna have these, you're gonna have temperature. Oh, look, it does TPU equals true automatically. I don't know if that would be necessary. I might set it to fit TPU equals false. Whole bunch of options, man, so many options. And then down here it describes what the arguments actually are. CO2 limit, T leaf is leaf temperature in Celsius. Oh, I thought it was supposed to be in Kelvin. Okay, it's supposed to be in Celsius. So the units is expects for CI, unit is expects for the light. Atmospheric pressure in kilopascals. Like there's all kinds of useful, lots and lots of information in here. And then it also has references for where it got the formula from, basically. Okay, and at the bottom, it has examples for how to do this. The thing here that is a little bit unique is you have this, you want to make sure that you're only using one light level for fitting your ACI curve here. So the first thing we're going to do is okay, so for our data set, so C3 ACI, we're going to add a Q2. Well, we can look at what Q1 looks like, right? So I'm gonna just type down here. And so it's about the same for everything. So you could probably skip this step, but if you had a sheet in which you measured the ACI curve and there was data at different light levels, you would want to exclude those. So to be perfectly, in an ideal world, to follow what the example says, we're going to do this. We're going to go round C3 ACI Q in, and we're going to round it to even, just to round numbers. So now everything's roughed up, is out, evened, rounded off to 1500. I don't think I have to convert it says to convert the leaf temperature to K. Okay, this is the part where we're going to use the UD units package. So T, and then I'm going to do, there's T leaf is already in here, so I'm going to do T underscore leaf. And I'm going to do ud.convert is the function. And we're going to convert C3 ACI T leaf. And then the first argument is what it actually is. So it's in Celsius. And we're going to convert it to Kelvin. So again, down here we can compare. So T leaf should be, yep, large numbers, whereas the T leaf without the underscore is much smaller numbers. So I did the conversion perfectly. Take some guesswork out of that. This is also very useful when you're converting fluxes and there's like per grams versus kilograms per meter squared or per millimeter squared or centimeter squared, or it can handle more complicated conversions, multi-step conversions in one step. Okay, I think, oh, and the last thing we need is to untibble it. This is a tibble, so I will show that. If we do str or structure on this object, well, first of all, it's long because it's got 153 columns, and up here is a tibble, and it's all, yeah, so we need to untibble it. So that's why we had the untibble function at the top. And I bet most of you could probably rewrite this into in the dplyr way, so where you pipe it. I just want to show it here in BaseR, and then we can also do it the piping way on the next time we have to do this. Okay, round Q in, convert to K, and that's a good note for that little chunk. Okay, finally, we're going to do a fit. That's the object, and we're going to use finally this fit ACI response function. First thing is data, right? So C3 ACI, and next thing is bar names. And here I'm going to refer to, it needs ANET, TLEAF, CI, PPFD, and I think that's it. I'm not even sure if it needs. Yeah. Oh, this is what it wanted us to do. It wants us to limit this for C3 ACI $Q2. Want to restrict ourselves to the 1500. Again, not strictly necessary for here, but it is recommended in the help file. So we're gonna follow that to a T. Bar names, A_net. In our case, that's called just capital A. Oops. T leaf. We called that T_leaf after we converted it to Kelvin. C_I. I think that's just CI. No underscore. And then PPFD equals Q. Well, I guess you could do Q in, or we could do Q2. I don't think that would make any difference, but you might type Q in here. because it doesn't matter. We restricted it with this rounded version. We restricted it, so it shouldn't matter. Oh, and I did say I wanted to do fit GPU equals false. Okay. That was a lot of typing. I'm going to go ahead and drop this into the chat in case there's some comma or something that's tripping things up. Whoa, that showed up terribly, but should work. We might have to format it a bit. Okay, so we can run this line, Control Enter. Doesn't take very much time. One thing you might check is like, what is the length of this object and what are the names of this object? Oh, so they have a fitted parameters, a plot, and a data. Well, we're most interested in the fitted parameters. So let's look at the first list element with the 2 square brackets, put in 1. It returns an estimate and a standard error of that estimate for these parameters, many of which we talked about: VC max, J max, RD. Oh, so they call it alpha instead of phi, 0.24, among some other things we did not talk about. So again, this is I'm showing it for one curve, but if you're running this for many curves, you would import them all in, do all this to one of do the same formatting that we just did, and then you could extract out a whole, produce a whole table of these parameters that are of interest to you. Okay, and then fit. This is nice. This is not in the Plant EcoPhys package. Fit2, it produces this curve, plots your data, so you can see, oh, the right point, the circles here are my actual points, and then this is the carboxylation limited, and then the electron transport limited curves. So I thought that feature was pretty cool. You could plot it yourself, of course, but it's nicer to have it be a part of this output. That's it for C3. I'm going to move on to C4 now. Is there any pressing questions? Okay. Guess I don't need to save this yet. Okay, so what did I type before? I said import C3 AI curve. Okay, so now we're going to import C4 ACI curves, ACI and AQ curves that were also taken off of 6800. And so I'm going to label them C4 ACI gets. And these were cleaned. So someone did go in through here and take off all the headings. But unlike the previous one, this one has multiple, so there were 3 different replicates. So there's additional columns here, unlike the first one. So we're still going to go read CSV. We're going to go inside the data folder, inside the ACI folder, and this one is called SEVI for Setaria viridis, or C4 plant, 2020-0304. Man, that was just before the pandemic got to a crisis level here and things started shutting down. One of the last things somebody did before they had to go home. Okay, so this has some duplicated columns, but if you look at it, it doesn't have the heading problem. We don't have to do the skip column names, skip rows, any of that stuff. This has already been trimmed by hand, basically. And the species, there's an additional species column here, which happens to be all the same for this one, but you can envision a data set where you were mixing your species on the same sheet. And then there's also replicates. So this one has plant 1, plant 2, plant 3. There's 3 plant replicates here. Okay, the reason I'm importing both is because the BioCro package, the OPC4 photo function, really needs both the light response curve and the ACI curve at the same time. So for here, we're going to do both. I'm going to do AQ, and I'm going to do copy and paste and just change ACI to the AQ folder. Read that in. And it has the same duplication warnings, but otherwise the data look, yeah, the headings are here, got species and rep, everything looks kosher. I will drop these two also into the comments in case the typing of the numbers slows anybody down. Okay, once again, plotting, can't escape the plotting. CI. Okay, oh, I should add a third one, color equal rep. Okay, so now we see the 3 plant reps. This first curve doesn't look that great, but again, I feel pretty powerless to distinguish between, you know, if I were the person who actually collected this data, maybe I would have more reason to exclude certain points and not others. But as just the user of the data, I don't feel qualified to throw out any points, but maybe the curves for plant 2 and plant 3 will be stronger or have less error than for plant 1. That's the ACI curve. And then we can do the same for the AQ curve. So I just copied and pasted and changing a couple letters here. Instead of CI, we'll plot QN. And this looks really good for both, for all 3 replicates. Okay, the function is in the BioCro package, and I'm going to query it as well, because, you know, first time you use anything, you got to look at the help files for it. It's OP C4 photo. At the moment, VC Max and Alpha are optimized only, and you would know, well, VC Max comes from an ACI curve, Alpha comes from an AQ curve, so really you need to give both sets of data for this to run properly. It is for the COLATS version of the C4 model. I'm sure there's other C4 models out there as well. And then it just says data, and it gives a bunch of starting values, but down here in the arguments, it describes what that data needs to be. So it's your observed data. It should be a data frame or a matrix, so not a tibble. We're going to have to untibble it again. The first column should be net assimilation rate. Second column should be quantum flux. It provides units. Third column is temperature in Celsius. Fourth, relative humidity in proportion. Optional fifth column can contain intercellular CI. Okay, so we need to make this data frame. Let's see, it's actually lowercase. So I'm going to just shorthand, call it DF for data frame. So I'm going to draw in pieces, and it needs to be from both those data sets. So what's the best way here? I'm going to go back up, and I think I'm going to make a combine. Let me make sure I can combine them. Should be able to be combined. They have the same number of columns. Hopefully they're the same. Rebind dot data dot frame before. The names do not match. What if we take like the first, like, let's see. Where do we need it? What's the last thing that we need out of here? The A and the CI are all pretty far in the front, but the RH chamber is also pretty far in the front. Q in, where is Q in? 71. So let's take like the first 71 columns, or so 75. Let's be on the safe side here and see if we can't get those to R-bind. And they did R-bind. Sweet. Okay, that's because we need to name things here. So we're going to do comb. First thing is net assimilation rate. The second column should be observed quantum flux. And this does not want us to convert to Kelvin, so we're just going to leave it in Celsius. Oh, I guess I don't know where tea leaf. I didn't look for where tea leaf was. It's tea. 84. So let's see if we can't do 85 up here. Rerun that and see if it matches. Whew. Okay. So it should work. And then fourth column, relative humidity in proportions. I'm pretty sure this is not in proportions. Let's look. Summary. RH chamber. Yeah, this is in percent, so we're gonna have to change that to proportion, divide by 100. So RH chamber, divide by 100. And CI is straightforward enough. CI. Huh, left, right. Here, look at list types. Anybody catching what mistake I made here?
I think that tea leaf
Oh.
doesn't exist in comb.
Oh, I think it may have been this. I had a accidental space between in my get symbol. You don't think tea leaf?
Now,
Oh,
if you check comb $, tea leaf is not one of the name of the column.
Ah, sorry. I went back and changed it to the first 85 columns.
Okay. Yes.
I had to figure out, I did a little to query which, there's just so many columns in those freaking 6,800 sheets. I had to figure out which one it was. It's the 84th, so I added in 85. So if you do that and rerun the comb line, then tea leaf should show up. But yes, I ran into that same problem. Okay, and fitting the call outs. So the fitting part is actually, that part is like not the hard part, right? The hard part is making sure you know what the column names are, making sure that the units are correct. The actual fit, I'm gonna just call this fit C4. The running of this is not the hard part because that's all I do. I'm going to leave everything else standard. You might wish to explore, the other settings for initial values, etc. A lot of these are initials, so it doesn't really matter what you do. It's just letting the algorithm converge, and then we'll check what this looks like. So this one, it's not like a list of many elements. It's just a single output. It gives you the 95% confidence interval for Vmax and alpha. Again, alpha is the same as phi from the presentation. And it shows you, okay, that's pretty low correlation between the two. And then convergence, yes, okay, it converged. It doesn't have a neat little like plotting feature or anything else, but it does give you an estimate and it does give you some kind of interval around that estimate for C4 parameters. Okay. We are getting to, it's 11:15. There's only 15 minutes left, and I'm a little bit sick of hearing myself talk. So I'd like to open it up. Again, the code is all available to you. So let me just walk through that. Under scripts, there's everything I did today, plus the only thing left so what we did today is found under script_photo. The thing that's underneath here would be how to fit a light response curve, and then how to do it for multiple individuals. So you note that for the C4, I actually combined all 3 individuals and just ran them at once. You could also split each rep out and use a for loop or something and estimate the parameters for each individual plant, if that's the level that you're interested in. And then the photosynthesis package has a way that lets you fit a whole bunch all at once, which is also really cool, without doing a for loop. So it's a function way, so it'd probably be faster than doing a for loop. This is the model script. We can take a look at that. This uses the more classic plant ecophys package, loads in some data and plots it, removes an outlier, and then there's an example of fitting Ballberry looning, and fitting Medlin, and the variables you need are slightly different. And then here's an example of the plant ecophys package also has a way to fit multiple curves. You would just have to give it this group. So what are you grouping by? Are you grouping by your column named replicate, your column named species? In my case, my column named shrub ID. And then finally, there's a Bayesian version from the Pecan. It takes in the example. I didn't want to make it too complicated. The example is for 6400 data, and it reads it in the ASCII format. I don't know how many people, I don't know whether you would say most people save that or not. And then you can do a fit A to the combined. It's kind of like the C4 example we just did. You would take those both curves, smash them together, and then you can fit the curves simultaneously with this fit A function. And you can look at the results. There's plots in the results. You can summarize the results. Like there's things that come out of the results there. So they all have a similar structure. And again, the hard part is not using the function, right? The function and all those equations, like the hard work, somebody already programmed that and hopefully double and triple checked it that it works under most cases. The hard part is getting your data into this analysis pipeline in a way that's consistent and reproducible, and that future you will remember what past you did in case there's any question that ever comes up, whether it's your advisor asking you or reviewers asking you to go back and check something, that you have something to go back and reference and check what you did. And with that, I think I'd like to open up for some comments or questions, or if there's any particular data, like if you have a sheet right now that you want to jump in on and try to do this on your own. I think I'd like to have that format. And again, Christina, me, and David can answer questions. We can go into a breakout room if you don't want to talk in front of everybody about your data. But an hour and 15 or an hour and 18 minutes is kind of a long time for your undivided attention. So thank you for that.
Well, I have a question.
Yes.
If I can. Yeah. So how many data points normally do you consider in these curves?
Oh, that's a great question. Well, we can look, right? What did that C3 have? This one had 14 data points.
Yeah.
And let's see also the order in which they do it, right? This one started about, this one, the CA started about 400 and it went down and then it came back up and it redid the 400 or the near 400 twice before going up. I've seen different people have just different preferences about this. We have one collaborator that insists on redoing this 400 measurement like 8 times before going up, because something about the way the enzymes are adapting to different CO2 conditions and how long it takes. That's her preference.
So is that a problem for the function? Because what I did like with the ACI in sorghum was like to start also from like 400 going down, then going up again.
Mm-hmm.
But I see that like at the same CO2 values, when you go to the curve down and then up, you have very different photosynthesis response.
Yes.
So do you have to clean, for example, the curve when you're going down, and then you use only the points when you have like the ramp up of the CO2, or you can fit both and the function is able to handle that?
That is a very interesting question, and it's both like a technical question of like what is the plant doing at that time, and like more of in some ways is also a statistical design question, kind of how can the algorithm handle variability about a certain point. it seems to me that the reason people do multiple, like especially when they're coming back, the reason they do multiple measurements at 400 coming back up is because they know they're going to throw out some of those points. They know that the first few of those, it's still the plant acclimating. So you could either use your judgment and say, I'm going to plot each one and look at it and remove X number of points, the ones that look bad, or you could have an algorithm that says the first, I'm going to run it 8 times at 400 and the first 4 I'm going to throw out, or the first 2 I'm going to throw out. I don't think that answers your question about like going down versus coming back up. I would assume that going down, like you start at 400, right? And presumably the plant under normal, if it's not in some kind of elevated treatment, is accustomed to 400. I think that first measurement is okay. It's just when you come back up to, from 0 or close to 0 up to 400, those first few measurements at 400, it's still acclimating. So make those first few when you come back. I think those are the ones you typically throw out.
Okay. And thank you. And can I ask you also another question? This function that you presented today, you said like you had like 14 measurements of data points in this kind of curves, but can they, because in the racer you have many more data points normally when you get that. So once you correct for the empty chamber, that is like all the tricky part of using like a racer measurements, you have anyway, it's very similar curve as a normal ACI, but with like hundreds of data points in the end.
Oh, wow.
So if I fit a curve with so like hundreds of data points into this function, can the function handle it, or?
Oh, I would say yes. I think especially if it's quality too, right? Does the curve look like a curve or does it look like a cloud of points?
No. It's
you have 100. It's
normally the racer is like just that you have like this ramp that you have as a kind of mismatch between the detectors in the Liquor. So you have like to correct for it, but once you correct for it, then it's simply a curve. Okay. But instead of having few data points, it's very, very dense, but it's not like moving. It's just linear. It's just a linear curve.
Oh, well, yeah. You could aggregate all your data, all your species or replicates of these little curves, these non-racer curves, and aggregate them all and feed them the function. And I don't think it might take slightly longer to run with more data points, but there should be no reason that it can't estimate exactly what we do here with 14.
Okay, thanks. I will try it.
So I guess I'll make one quick comment, which is that my understanding is that the person who developed the racer protocols, working in collaboration with Liquor, they also wrote an R package to fit those curves. So, I'm not very familiar with it, but I would defer to that package. I think it only works for C3 plants. I'm not sure exactly how to adapt it, but on the topic of rapid ACI curves, one thing that I noticed about this curve that Jessica has in particular is the fact that the data points were taken pretty quickly relative to my recollection. The key is that at each step, it's necessary to make sure that the values stabilize. So I can't remember if that's more important with the light curve than the CI curve, but yeah, I guess making sure that you give the time for the leaf to equilibrate is that's essential. And then ideally it'll reach the same values at same values of photosynthesis at 400 that it had when it was out there doing its thing in ambient air. That's especially for modeling, that's kind of the idea, is that you would have that this would match what's happening when you step up to the plant in the first place. So
okay.
How long does it take to get that very dense hundreds of points curve?
Well, I think that the idea is that actually the equilibrium, it's like a dynamic process. In that case, it's not really that you have steady state at every point that you set for the curve. But I have both. So I did like on the same plants. I did first ACI, and then I alternated to a racer. And the racer has a ramp that I think it's the one that works the best for us was like 100 ppm per minute of ramp. And then you just like log data every 2 seconds. And the only problem that you have is that like the two IRGAs, like the one before and after the chamber, they are like mismatched because of these continuous flows of CO2 that you have inside the chamber. So you have to run, before running the racer curve with the leaf, you have to run a racer curve with an empty chamber, so you can see how much is the mismatch between the IRGA before and after the chamber. And then you use that curve to correct your curve with the leaf in the chamber. And by that correction, you avoid like negative numbers or stuff like that. And you get like a pretty similar curve that is just very densely fitted with data points, because it's every 2 seconds. And then like, yeah, there's a package that can handle that for C3, but not for C4. So I asked this question because I wanted to try if the OPC4 photo function can actually handle many data points as the one that I have in Eraser corrected already for the empty chamber effect, and see if I can get like similar estimation of Vmax between the normal ACI that I ran before and the Eraser that I ran after. Something like that.
You know, one thing I think you might run up against is I tried running that with just the we combined the ACI and the light curve. I tried running with just ACI, and it doesn't converge with just the ACI curve. Do you also have light curve data?
No. No, we don't. It's just an ACI curve.
So another question about that. So then is the key, if you're looking for VC max, you're looking for the asymptote of that curve, is that correct?
Yeah.
And so it might be straightforward to simply fit fit that asymptote and with a different modeling function.
Yeah, you could write you could also dig, all of these are open source, right? I don't actually know what I'm going to find after typing this in. But but at its base, it has some kind of fitting, nonlinear fitting NLS or something like that in there, and you could fit your own model form and estimate VC max from your dense data. That doesn't, but you might not be able to use this package function.
Okay.
Does anybody else have any concerns, detail, technique involved, either on the technical side while we have everyone here, or something about the code?
I have a question. So I guess 2 summers ago now, we were trying to do some VPD curves. Has anyone had any success doing those? We were not able to get the data to stabilize to be able to log values.
I think the answer is no. From what I've also dug into this because I was really curious, the STIMADL models, if I can go back to the a thing that's kind of almost annoying about the 6800s is that now a lot of people collect data with VPD set points, right? They set the VPD to like 1 or 2, and then you're fitting something like a Ball-Berry Looning or a Medlin, where the only variability is coming from the photosynthesis varying, but not from the VPD varying. And kind of the more fundamental, like when you fit something, you need variability in the X in order to fit a line or a curve or something. If you just have everything at a single VPD, that's a super narrow range, it's really hard. Like you might not get convergence, or you get nonsensical values sometimes. So yes, the long answer is or the short answer is no. I don't know if think people have done it successfully. I think people are trying, and people understand that this is a problem to collect things at a single VPD and then be able to expect to fit a good STIMADL curve to it. But stabilization is a problem. So I wonder almost if instead of a curve, it might just be combining data from the same plants or same leaves on different days when the VPD is just different. So measuring, yeah, doing spot measurements on a wet day and on a dry day and on a medium day, combining try to account for as many of the other variables as you can, combining those and seeing if you can fit. And for me, always the stomatoconductance conductance curves are always way messier than ACI. Like ACI, like you take, like it looks like a curve. Stomatoconductance, if I were to plot like the Ball-Berry, for example, and make it linear looking, it's kind of a thick cloud of points. It's not a super tight line by any means. David, did you want to chime in on that one?
No, I was actually just looking up, I apologize. I was looking up the, for C4, whether or not VP, like what the value of VC max versus VP max is. So I'm totally on a tangent.
That's okay.
but I think like the rate limiting step, the asymptote is VP max as opposed to VC max. But I was looking that up so that I could talk without speaking incorrectly. So I don't want to diverge.
And Kinsey, if you ever want to talk about this further, I'm kind of interested in like the whole, like seeing what would it take to get a, maybe not a stomatal VPD response curve, but some measurements in which VPD is actually varying to see if that better fits, and you get almost different estimates of parameters than if you just collected measurements all from one VPD setting on your 6800 and tried to fit curves to it. I bet the answer would be somewhat different, especially for this Ball-Berry looning, when you have to, if you're trying to estimate this D naught and there's no variability in D. I mean, I feel like that's just a impossible, like you'll probably get a number, but it's not right.
Yeah, I agree with that. I will keep that in mind because I might be working with some of that data here in the near future. Even though we didn't get any VPD curves, I'm still interested in kind of the whole issue around that.
Yeah, it definitely seems very necessary. Hey, it is past time, and I know that everyone's busy and I don't want to keep you guys over time. Christina or David, could you drop the office hours link one more time into the chat? And just so Tuesdays between 8 a.m. and 10 a.m. we're on a gather town for ResBaz's Coffee and Code, but we co-locate with them in virtual space. And we're just little avatars in a game space, and you can walk up to us and chat about your data woes or life in general. So, okay. Well, I'm gonna probably stay on here with Christina to do some do some housekeeping, but otherwise, thank you so much for attending. Again, all of this material is on the GitHub repo. We are going to continue working on getting that binder instance up so it can work, and we'll send a follow-up survey with some links, and yeah, and then your survey responses are much appreciated.
Thank you, Jessica.
Thanks.
Yeah, great job. Thanks.
Bye-bye.
That was awesome. Oh my
Yeah.
gosh. You picked, like, you were, even though like everything wasn't quite correctly prepared, you did a really great job of just like making it work and moving forward. So yeah, I thought that was awesome.
Oh, thank you. I'm like so sympathetic to the like desire when you're talking to no response at all to just speed up and keep going faster and faster. I'm like, don't do it. Don't do it. But this is so awkward. Nobody's saying a word and they're just staring at me.
Yeah, I've been having to practice that very intentionally a lot. Like when I say, does anyone have any questions? I'll try to sit. I should like have a timer or something where I can set it for like 30 seconds or something. Because even just unmuting yourself to ask a question or like type something in the chat takes a second, you know? So, but it is so awkward, especially when no one's video is on and you're like, during the workshop yesterday, I was like, am I speaking into the void? Like, is someone out there? They were still all out there, but I use those thumbs up reactions a lot. That's really, really helpful because it gives, it's like the red and green stickies, you know?
Yeah. But yeah, this online teaching takes some getting used to.
Yeah, and sorry about the technical difficulties.
It's not your fault. I was there for the binder instance that worked, and then suddenly you made it less complex and it didn't work, and I don't know why that would be.
Yeah, I mean, apparently just there are problems with, like, they discourage putting Docker containers into binder instances. And sometimes weird things will happen, and they're not willing to post like the logs from binder itself for whatever
Wait,
reason.
you're not supposed to put Docker files?
You can, but it's like it should be your last resort. But I'm not sure how you can have an interactive RStudio session without using Docker. Like, you can use it to set it up sort of like automatically run your code on R or whatever. And it is set up for Jupyter Notebooks, which is interactive. So however I don't know, I need to look into it a little bit more, I think.
Oh, yeah, I mean, I think they do provide the RStudio whatever stuff, and there's just a URL parameter that's something like that says RStudio to launch it in RStudio rather than Jupyter.
Yeah, but the only way that so I was basing how I built it or set it up from Phenome Force, and they set up a Docker container to get the RStudio instance. And so there's maybe a different way to do that where you don't need Docker. Maybe that was just the way that was easiest for them. But I think if it's possible to have that interactive RStudio interface without Docker, then like I'm going to try to figure out if that's possible.
I thought it needed Docker. I needed a Docker, especially to get like the JAGs and like all the more complicated stuff. Because it's, we're not just for that stuff. It's like, it's not just R, it's JAGs and crap.
You can install things through the command line in these binder instances. It's just more difficult. I will poke around and try to get this to work during this sprint, but I don't know. We'll see.
Hey, David, I was trying not to say anything incorrect, but you said it's not VC max, it's VP max for C4?
Well, that's why I honestly, I always get confused about this. But I think that VP max is a rate limiting step for like it's a CO2, when you get like that asymptote is limited by whatever this other enzyme is, because the carbon concentration
isn't limiting.
Yeah, I mean, so the carbon concentration mechanisms limit the or make sure that the carboxylases always has enough carbon, and it's this other thing that I don't know what it is, makes, what's this thing called, PEP carboxylation.
PEP carboxylase.
That limits like how quickly it can use that CO2.
Okay.
Yeah,
yeah, this feels like it just shows me how little I actually do know about photosynthesis. And even just putting this together felt like it required